Hi, my name is Paul Battlay. I'm a postdoc in Hodgins lab at Monash University, and today I'm going to be talking about some work we've just started doing in white clover with the Global Urban Evolution Project. So white clover is an agriculturally important forage crop that's native to Europe and Central Asia, that has been introduced to all other continents across the world except for Antarctica through human activity over the past approximately 400 years. And white clover is also an emerging model organism for ecological genetics, thanks in no small part to work by this Global Urban Evolution, or GLUE project, who had this paper earlier this year where they assessed parallel patterns of urban-rural evolution in 160 cities in white clover. And in doing this work, they resequenced approximately 2,000 white clover whole genomes across the native and invasive ranges. And these data sort of suggest that there's been extensive admixture during these invasions, and they also show 5 genetically diverse and distinct introductions in North America, South America, Oceania, China, and Japan. And as has been said many times, we can think of these as replicates for natural experiment, where evolution begins anew. And this just provides a really good high-resolution data set for understanding the genetic architecture of invasion. Specifically, we can ask whether we can identify genomic signatures of adaptation following invasion, and whether we're seeing the same genomic regions utilized in these distinct invasions. So what are our expectations of invasion and its genetic architecture? So we can observe that adaptation following invasion often happens rapidly as haphazardly introduced invaders spread across diverse environmental gradients. We see the re-evolution of trait clines that are observed in the native range, but we also see response to novel environmental pressures. We also expect that invasion and rapid adaptation will involve large effect genetic variants. So there's this theoretical work by Alan Orr where he sort of demonstrated that following a large environmental shift, populations may take large mutational steps to return or to readapt to this new optimum, particularly early on in the readaptation process and their adaptive walks. Now, additionally, large effect variants are resilient to stochastic loss, which may occur when population sizes are small or when gene flow between locally adapting populations is high. And one source of large effect genetic variants are chromosomal inversions, which are these inverted sections of chromosomes that we've already heard about today, which, when heterozygous, suppress recombination. And this allows them to collect and combine multiple small effect variants and segregate as a single locus of large effect. And there's substantial theoretical and empirical evidence to suggest that over time, some inversions may evolve into cassettes of locally adapted genetic variation. And given the inversions of these dramatic genomic changes, they're actually really challenging to identify, and this is because most population genomic samples are resequenced in small pieces and aligned to a reference genome, and this is really good for identifying SNPs or small changes between individuals, but it's really uninformative for large-scale rearrangements. But some inversions leave footprints in population-genomic data in the form of variation in local genomic population structure. So we use this method called low-struct to see if we could identify inversions in our white clover data. And just looking at this method in detail, we started with the SNP genotype likelihoods across almost 2,000 clover genomes. And then we walked along each chromosome, performing PCAs in each 100 kilobase per window. So you can think of each of these points here as representing a PCA. And then these PCAs are summarized by multidimensional scaling. And the way we interpret these MDS values is that values close to 0 are kind of representing the normal population structure observed along the chromosome. What we're looking for in identifying inversions are groups of windows with divergent local population structure, sort of that we see in extreme values on this MDS axis, so things like this. We also expect that the genetic variation in an inverted region will cluster into 3 groups on one axis of the PCA. So, and these would be representing the 2 homozygous and 1 heterozygous genotypes of the inversion. And then finally, we'd expect that the average local heterozygosity at the nucleotide level would be elevated in the putative inversion heterozygotes. So higher heterozygosity in our putative heterozygotes compared to the homozygous classes. And across the Clover genome, we found 2 regions that ticked all of these boxes on chromosome 7 and chromosome 8. So 2 strong inversion candidates. And I will come back to the inversions, but I just wanted to talk about our analyses to detect genomic signatures of adaptation following invasion. So we ran this Baypass contrast statistic that's been bandied about a bit at this conference already. So basically comparing population allele frequencies between 2 groups of populations. In this case, we're comparing native and invasive. And it provides a null model to account for complex evolutionary history, so basically a correction for population structure. And we ran these scans, 5 pairwise scans, comparing Europe with each of the invasive ranges individually. Right, so the results of the scans shown here, there's Manhattan plot, so there's the contrast statistic on the y-axis, negative log transformed, and then the genomic location along the x-axis. Now we also analyzed this data in 20 kilobase pair windows to leverage the fact that selection will likely drive multiple SNPs in a region. Windows that are enriched for outlier SNPs, the top 0.5% of these are highlighted in red, and these are sort of like candidate outlier windows. And just like eyeballing this data, we can see that there are like several regions which appear to be shared between these invasions. And yeah, just looking at this question more quantitatively, the bulk of these outlier windows are actually private to each individual invasion. However, there is substantial overlap between these invasions, and if you look at these invasions in a pairwise fashion, this is far more sharing than would be expected by chance. So this is some really strong evidence of genetic parallelism in these distinct invasions. And then if we return to our inversions and plot them out in the genome, they're indicated with these blue bars here. We can see that they're associated with extreme outliers, particularly in North and South America, and particularly the putative inversion on chromosome 7. Now, given this idea that inversions will collect genetic variation that is adapted to a particular role or environment, we looked at the enrichment of biological processes in the gene annotations in these inversion regions. And there are a lot of geoterms that look like they'd be useful in colonizing a new range, but in particular, these pathogen defense geoterms, which pop out in both regions. And this is a really attractive hypothesis because we expect pathogens to be one thing that varies between a native range and an invasive range. And what we could be seeing here is either a response to novel pathogens or an escape from pathogen type thing where genetic resources are reallocated away from defense. So we've seen that we can identify genomic signatures of adaptation following invasion in white clover. And within these, we see strong signatures of genetic parallelism. And this implies a lack of redundancy in the genetic architecture invasion in the species. We also see the involvement of putative inversions. And We can ascribe a potential role to these, which is the changes in selective pressure from pathogens. There's a lot more we want to do on this in this work. Up next, we want to partition the roles of local environmental adaptation from invasiveness. So look at genetic environment associations, for example, climate pollution. We're also really interested in the evolutionary history of these putative inversions. So Clovers and allopolyploid, and chromosome 7 and 8 are homoeologs, which means they share an ancient ancestor chromosome. So what we could be looking at here is a single inversion derived from an ancient event, but there's also the possibility that we're seeing a pattern from a more complex structural variant. So I'd like to thank the Natural Sciences and Engineering Research Council of Canada, Canada Research Chair and the Inversions Hub for organising this meeting and having me to talk. And I'll take any questions.
Awesome. Thank you so much, Paul. Does anybody have any questions?
Hi, it's about the inversion mechanism, I guess. Is there anything interesting at the breakpoint? Because some of the insect inversions, there seem to be particular regions of chromosomes that are predisposed to those kind of. So is there anything there that might explain why you might have a parallel event?
Yeah, that's a really interesting point. I'm not sure. We don't have a good resolution for identifying the breakpoints yet, partially because we're not using SNP data for the identifications. But that's definitely something that we're interested in. Like, it may not be a single event. It could be just that this chromosome is predisposed to it.
Anybody else have any questions?
So this might just be because I don't understand, but the PCA that groups the homozygous, the heterozygote, and the into 3 groups. Why is there stuff in the middle? Like, why aren't they more distinct than they?
Why aren't they more distinct?
Yeah.
Because my sort of thought on it anyway is that recombination isn't entirely suppressed in inversions, and it tends to be more suppressed towards the breakpoints.
Okay.
So you're capturing, there is some recombination in there, possibly also some noise as well.
Okay. Thanks.
Was there anybody else?
This may be a quick answer because I am very much not a plant person, but I noticed you put up the list of genes and it had a lot of microbial associations. And I'm curious, is Clover one of the species that, doesn't it nitrogen fix? Does it do that through a relationship with the soil microbiome, or am I showing my marine ignorance here? Because I wonder if that might be another potential driver of some of those signals, if that is indeed happening.
Yeah, it's possibly. I'm actually not sure either. I'm not really a plant person.
Cool. Thank you. It's always interesting to see that kind of array of notation, try to figure out what it means.
Yeah, and it's nice to put them in front of big groups of people because people point out different things in different areas.